Good afternoon, everyone. Welcome again to my presentation. I will be presenting on behalf of the quantum computing team at Roche Pharma. And today I'll focus on the different machine learning use cases that I've been working on in the context of the pharma drug discovery pipeline and how we try to apply quantum computing to the mix. So yeah, a rundown of the agenda for today's presentation. I will start to give a general overview of how Roche Pharma is structured and how PRED fits into the picture. I'll try to also explain why machine learning is so ubiquitous in the pharma context, and deep dive a little bit into, as promised, how we apply quantum computing into the picture, and end on a very brief note on the other quantum computing related works that we've been doing. So without further ado, here is the schematic of how Roche Pharma is structured. We have 3 independent R&D research and early development units. PRED is one of them. We're based in Basel. The other 2 is based in San Francisco, very close by, and 1 in Tokyo. So the 3 of us share one global function for late stage and commercialization. And by late stage, we mean clinical development. So what does all these concepts I just talked about mean in the pharma pipeline? What does R&D mean in pharma? So we start with preclinical research. So to make a drug, we start usually with what we call the therapeutical hypothesis, namely that we need to determine a target, usually a protein in the human biology, which is responsible for some kind of function. So to make this example concrete, this can be headache that we want to eliminate. And we need to find some sort of receptor that we can modulate and that can help ease the symptom. So with this specific example, we've identified, for example, the cannabinoid receptor, and that has shown to be very effective in modulating headache. And so once we have a working hypothesis with a right target, we need to move on to a lead identification. So this is what I would call reverse engineering, where we have a target and we need to be able to find a molecule that can modulate this target, so inhibit its function or amplify its function effectively. And usually in order to do this, we need to screen thousands and upon thousands of molecules to find the right one that can modulate the function with the right level of efficacy and not too many side effects. Once we have narrowed down to a panel of molecules that can target our protein, and we're fairly confident with its behavior, then we can move into what we call clinical development. So this is where the molecules are tested in humans. And with each stage of clinical development, the molecule needs to pass through a certain set of criteria. In phase 1, we usually need to test for safety and the correct dosing. In phase 2, we're looking at the efficacy in a small group of patients. And in phase 3, the molecule is pitted against a larger group of patients, and it's usually tested against the standard of care. So all this needs to happen before we apply for approval to the regulatory bodies to commercialize a molecule which has, by this stage, been proven with statistical significance that it's better than the standard of care. So this is a very long, drawn-out process that usually takes more than 10 years. And I would like to now move on to highlight for you how machine learning plays a role in each step of the process. So starting with target identification. So again, here we're looking for, let's say, the needle in the haystack, where we're navigating through very complex human biology to find that one protein that can help us modulate some kind of function in the body. And so traditionally, scientists have been doing spending a lot of time on understanding the disease and mapping out pathways. So more recently, we complement this with a more data-driven approach. So this means that we can now, with machine learning, mine through large volumes of genetic information in large populations and to help us quickly identify targets. And another example in this space is the protein folding problem. And here it's probably a more famous example as made evident by the AlphaFold algorithm. And there the problem is that we're trying to predict the 3D shape of a protein given its sequence of amino acids. And if we can do this successfully, then can really speed up the process of finding the right druggable site on the protein where it is a suitable target for us to focus our attention on. And so moving on to lead identification, here again we're optimizing our molecule in a high-dimensional space. We're profiling the molecules not just in terms of efficacy and safety, but also other pharmacological properties. And so if we do this, let's say in a brute force way, then we are screening simply every single sympathizable molecule in our library. So an approach that we call high throughput screening, and you can imagine this can be highly time-consuming and expensive. So the goal here is to speed this process up with computer simulations where we can predict, let's say, based on previously profiled chemical structures, for new chemical structures that, based on similarity search, we can predict all the list of properties that's shown here on screen, and thereby reduce the need for screening. And very interesting, we can also do generative AI, and we can generate novel design of molecules by using reinforcement learning. So plenty of opportunities here. And last but not least, in the clinical development phase, the ultimate goal here is usually to optimize the success rate in each of these trials, because each of these trials are rather expensive to operate, and we do want to make sure that we have the highest rate of success possible for our patients. And even despite our best efforts so far, to identify the most druggable target and to optimize our molecule, because due to the complexity of human biology, response to treatment in the population is still very heterogeneous. So within the same disease indication, patients can have different levels of response to the treatment. So the ultimate goal here is to be able to stratify patients based on some kind of biomarker so that we can target patients who are the most likely to respond to our molecule instead of treating people point blank. So that is a quick overview, and hopefully to help you guys understand the various opportunities for machine learning in this pipeline. And now, sorry, the order of the slides is a little mixed up. How can quantum computing help in this regard? So as I hopefully convinced you, machine learning is really important, and I would say the most prominent algorithm in machine learning space is arguably neural networks. And here with medical imaging, this is a case where this is a flourishing area of research, and a lot of the most competitive state-of-the-art algorithms in the classical deep learning community is constantly pushing the boundary forward. And so this is also an area we have plenty of data in-house. We have very rigorous benchmarks available in-house as well as externally with open data sets. So when we're looking for advantage coming from quantum computing, we really wanted to pit the quantum computer against a very high bar, which is the deep learning algorithms available for imaging. And for the next couple of slides, I will be showing you guys results based on this MedMNIST, which was also used in our last presentation. And it covers quite a nice variety of different image modalities coming with the classical benchmark results as well. And so the goal, again, is to investigate whether quantum computing can really offer the potential to improve either the efficiency or the accuracy of deep learning algorithms. And together with QCware, we embarked on the journey to find two quantum architectures that has been shown to have superior scalability. The first one is the orthogonal neural network. So this is a fully connected layer which takes advantage of the fact that we can have very efficient subroutines to calculate the inner product very efficiently without introducing too much overhead to do the data loading. And since unitary is implemented on the quantum circuit is also orthonormal, that means our result is also coming with a nice property that we can have orthogonal weight matrices, which from the classical machine learning community has been shown to help with training of models to converge faster and to avoid vanishing gradients. And so all this packaged together, we were able to find circuits that gives us a modest quadratic speedup. And then in the second piece of work together, we looked into the famous transformer network, which earlier this morning we heard plenty about. So this is really the architecture that has been taking over the field in text and image and also other data modalities. So it's let's say in a nutshell, the power of this architecture comes from the attention mechanism, which is able to weigh each component of the input data in its global context, whether it's a token in your text or a patch in the image. And so we were able to find a way to implement a quantum attention mechanism, which is exponentially faster. And, oops. Here is an overview of the results. So as I mentioned, we used the MET-MNIST, and on all 12 data set, we benchmarked our proposed solution against first the vision transformer. So this is the original classical transformer architecture. Also on the second row here, you can see implementation of a quantum architecture without the attention mechanism. And then the three following rows are our proposed approaches. And you see that we can reach nice comparable performance in terms of accuracy and AUC. And what we like to highlight is that while the performance is competitive in terms of accuracy, our architectures do require much less trainable parameters, which confirms our theoretical prediction for scalability. So that was some results on a very nicely curated set of images. But we wanted to also take this up a notch because while MET-MNIST is nice, it comes with nice benchmarks, but that's hardly what we would call a problem statement coming from the real world. So I'm really happy to say that we've continued our collaboration with QCware, and then this time we're bringing on academic collaborator from Harvard, Professor Rosen, and together we're doing a project on MRI, so magnetic resonance imaging. And there we're aiming to investigate the feasibility of applying quantum machine learning techniques to help us in the reconstruction of these low field images and also, again, to image classification problems. And we're also interested to look into very state-of-the-art techniques such as MR fingerprinting. So the methods that we're looking at here is not only limited to supervised neural nets, we're also looking into other linear algebraic approaches, which I'll touch on very briefly in a moment. And on the right-hand side, you can see the team setup, and I would like to just mention that this project is funded by the Wellcome Leap Foundation, and there's a panel right after this talk to give you guys more details. I won't spoil any of the surprises there. So here you can see the schematic at the bottom of how this process of MR screening works. So you usually get the raw image from your machine, and you would like to do image reconstruction to enhance the signal to noise ratio. And then with this enhanced image, you can do classification. So for example, here we're looking at the brain scan. So this could be tumor or no tumor. So this process mirrors what happens in the clinic. And on the top, you can see that we try to map the different classes of algorithm and how it can be applied to each step of the process. No surprise, deep learning can be applied to both parts of the process as well as the end-to-end image classification problem, while the other two approaches, we're looking to apply it to the two separate parts of the process. So this is the last slide on this project, which I find super interesting. So I already took a little bit of a deep dive into the deep learning methods. And here with the, let's say, linear algebraic methods, we are looking into the determinantal point process, which is a model that was actually originally proposed to model fermion behavior. So you can argue that this is a method that's natively quantum. And we get the nice property from this method that given a fixed number of data points, we're guaranteed to sample the points that give us the largest volume in the spectral space. So what does this mean in terms of MR images? This means that for undersample images, we're guaranteed to pick out the data points that give us the most information. So this is helpful for image reconstruction. And this method comes with a nice property that it gives us reduction in model variance, and it generates a so-called unbiased estimator, which makes it very nice for downstream classification tasks using, for example, SVM and other tree-based methods such as XGBoost. And moving on to subspace learning, this is a related idea where we, again, looking at undersampled MRIs. This MRI, when you do a Fourier transform, you look at it in the K-space, happen to lie in the lower dimensional space and gives us a low rank so-called Hankel matrix. Please don't ask me what that actually means. But we can then do a nice matrix completion technique on this low rank matrix, and then this again help us to do image reconstruction effectively. So to be fair to the rest of the team, we don't only focus on machine learning, we also look into chemistry, simulation, and optimization. To no surprise, these two are all very important applications in the pharma space. So very quickly with chemistry simulation, we calculated the binding affinity of a number of different molecules to a very relevant site. And we were the first one to do this on the real quantum hardware. And then optimization, as I already alluded to earlier, we were interested in protein folding and also conformation sampling. So yeah, thank you very much for your attention. I hope there's still maybe time for a couple questions.
Already a question.
Yes, thank you for the presentation. Can you please comment a little bit on, for your quantum neural networks or other quantum work you're doing, comment a little bit on how you're doing the data loading. Is that something that's pretty elementary at this point? Are you using tools built by QCware or someone else? Did you invent something? And how are you getting the data out? Is all that easy to do now or still pretty challenging?
So I think the data loading part of the circuit is very much integral to the whole circuit where we do, for example, the inner product estimation. So it's kind of part of the package because I think a lot of, let's say, machine learning related algorithms that's proposed doesn't usually come with the data loading part or assumes the existence of a QRAM, for example, right? So that's usually has been identified as a bottleneck, potential bottleneck in implementing machine learning in quantum computers. But indeed, in our approach, we design the circuit in a way that it doesn't add additional overhead, so which you could expect in other algorithms. Hopefully that answers your question.
Thank you. Do you have any comment on how you're getting the data out as well?
I'm not really quite sure. Maybe which part, can you maybe be a little more specific about? Yeah.
I'll catch you after the presentation and ask a follow-up. Thanks.
Yeah, thank you.
Thank you for the question. We have time for one more question. We have time for one more question as well.
Again. Okay. Yeah. Okay. Thank you again for the presentation. It's not really a quantum question, but for low sample MRI or other images, you usually use compressive sensing. Are you using compressive sensing to amplify and select the optimum frequency components?
Yeah, so we're looking here at different types of technology. So as I mentioned, we do look into low field images, but we also look into, I would say, more state-of-the-art techniques such as fingerprinting, which is this multi-parametric approach that has even faster acquisition time, and that allows us to profile different properties simultaneously. So yeah, we're looking into different techniques.